cell culture. What's the difference between cell culture, tissue culture, and organ culture? What's the differences? Can you tell me? Tissue culturing, organ culturing, or cell culturing. These three are different types of culturing. Okay, keep that in mind, and I'll tell you the answer somewhere.
Tissue culturing and
Organ, O-R-G-A-N, organ culture, and cell culture. They are different. Okay, keep that in mind. I'll tell you the answer later. So what is the why we have to do the cell culture in vitro? Do you know what is in vitro?
in the lab.
What is in vivo?
It's also in the lab, but in vitro is more like using the in vitro is using how to say that? Using the cell, using the things that is outside of the living things, but in vivo is using the living things itself.
An animal or a human body,
Animal.
right?
Yeah.
So if you are doing a clinical experiment, most of the time it's in vivo. So what is ex vivo? Or ex vivo. EX.
Ex vivo is using the animal or the model but outside of the laboratory.
No.
No?
Is for example, I will take some part of your cell from animal or human body,
Yeah.
and then I do in vitro culturing.
Yes.
For example, I can do virus infection or do a gene transfection or genetic engineering. After culturing for a while, and I put this bioengineered cell back to the same animal or same person. That's ex vivo. I mean, the part of procedure is outside the body, but at the end of the experiment or at the end of the protocol, this tissue or this cell must be transferred back to the same animal or back to the same body. Could be the same species, different body. You can do that too. Okay. So why do we do the cell culture in vitro? There are advantages and there are disadvantages. The advantage is we can work on a single cell type population, and that means all the genetic characteristics are the same. You don't have to worry about, oh no, I have to deal with epithelial cell, I have to deal with the dermis. Cell culture is a single cell type, and you can do it at control condition. Everything is cultured in a dish. So you are the person controlling what do you want to feed for the cell. You can put a lot of the nutrient, you can put minimal nutrient.
Yeah.
And you can make it hungry. So you control the condition. And then superb access means it's easy to manipulate, meaning if I wanted to proliferate or make a lot of these cells, or I want to do any type of experiment, it's easy for me to deal with this cell, because all you have to just open the lid, that's it. And it's you don't have to dissect or homogenize like a tissue. So it's very easy to work with the cell. And the disadvantage is not all like an animal. Well, all you can see is a monolayer cell, and the physiology of the cell in a cell culture is just the cell. You won't see the cell in this culture interacting with other type of the tissue or different type of cell. And so that's the difference between cell physiology or human physiology. So you won't see the whole big picture of the physiology. And the environment is not like in vivo. I mean, this environment, the medium you put in the culture is something synthetic and something defined by the experiment, which is very different in a body. And the effect of serum plasma, that is, these are the supplement we added in to the culture medium. And most of the time, if you are culturing a human cell, but you are using a bovine serum, and that's the interaction between a human and bovine serum. It's not exactly, or the environment is not alike, and there are side effects from a different species serum. So these are disadvantages if you wanted to use cell culture to do your experiment. You have to think about all these shortcomings. Okay. And cell culture, you can do it in terms of research. You can understand the function of the cell. Basically, that's most of people doing it. And the second popular research focus is to study the toxicology before anyone want to do an animal test. If you can see the effect in a cell culture, then you are able to predict the effect that's possibly if you feeding to the animal or if you are applying to an animal. The second one is to do the tissue engineering. These days we are able to build up a skin-like or liver-like structure in tissue culture. Well, in our lab, we were able to do the bone and cartilage too. And by doing this tissue in vitro, it allows the doctors to do the regenerative medicine. That means fixing patient with damaged part. The third one is also for research. There are monoclonal antibody or enzyme, hormone, vaccine, not research, sorry, therapy. I had to change this one. Therapy, treatment. We can use monoclonal antibody to, what? Do you know what's monoclonal antibody?
I forgot, but I know, but I forgot, I think.
What is the difference between polyclonal antibody and monoclonal antibody? What's the difference? Is this molecular biology? It's not. It's more like cell biology. I think I have a PowerPoint. Let me check. I do have a PowerPoint for monoclonal antibody. Wait for a second, but it's in Chinese. Yeah, I just. So to obtain a polyclonal antibody is very easy. All you need to do is purify the protein you need. It doesn't have to be one single protein. It could be serum, could be a tumor cell homogenate, and then you inject to a mouse for 3 or 4 times, just like how you do the vaccination. After at least a month, you can draw the blood and the serum or the blood sample from this animal will have the antibody specifically to that protein or that group of protein. However, the antibody has a lot of different affinity or epitope to the protein you inject into the animal. For monoclonal antibody, the same animal, you obtain the spleen cell from this mouse. and then fuse with a myeloma cell.
Yeah.
Myeloma cell is a cancer cell, and what it does is this spleen cell, which contain specific cell that secreting a single apoptosis antibody, will fuse with that cancer cell. What it does is that these two cells membrane will fuse together, and the two nuclei will fuse together too. After fusion, this heterokaryon cell is the monoclonal antibody, but it will all mixed in one tube. The cancer cell allows the spleen cell to live indefinitely. So you can have a cell culture that, you can pass for more than 100 passage and then get a lot of antibody out of this cell culture. That's monoclonal antibody. But it takes a long time to separate these single cell colony from this mass. It took me 2 months to do that. That's another story, okay? So let's go back to why we do the cell culture. And we can also design experiment to allow your cell culture generate recombinant protein for specific diseases, enzyme or hormone, or you can do it as a vaccine, okay? So these are 3 major criteria that we need to do the cell culture. So today I will talk a little bit about the history of cell culture, just a little bit. And then we'll look at the equipment and technique for the cell culture. And these two I'll put them together to introduce what do you need in the lab and what is the procedure for doing a simple culturing. So the first person to do the cell culture is this Dr. Rocks, William Rocks, in 1880, long time ago. What he did is he take embryonic chicken and then put it into a saline PBS. So this will be like an organ. An embryo is an organ, and then like organ culturing. And then he can observe the heartbeat of the chicken embryo development just for a short period of time. But that's the purpose of doing this in vitro culturing. And in 1900, Dr. Harrison, he developed a way to grow the organ or the cell by hanging down like this. So he grew the tissue, not a cell, on a cover, look like a cover slip. Then you hand over, turn it down, and then put the medium in here. But the reason he did that is because he wanted to observe the cell type under the microscope. By culturing like this, he was able to observe the cell morphology under the microscope. That's why he did this, turning down procedure. And he also found all these cells are anchorage dependent. You got to do adhesion here, otherwise it's going to drop into the medium. However, the growth rate for these cells are very slow. So probably take 2 or 3 days to see cell doubling. And for bacteria, the cell growth is about 15, not cell. Bacteria growth is about 15 minutes to 20 minutes per cycle. For most of the mammalian cell, it takes at least 1 day. Except for some stem cell, it takes about 18 hours to 20 hours for stem cell to do the replication. And he also found it's very easy to get contamination, and that's the main issue for doing the cell culture. You need to try to keep away those possible contaminants. Otherwise, bacteria will go in here, your fungus will go in here. You're not able to see the cell you are interested in. In 1900, at the same time, Dr. Carroll, he, in order to get away from the contamination, he designed a culturing apparatus, the flask, called Carroll flask. It's this one here. This is the flask. We call it tea flask.
Tea flask.
The 25 tea, because 25 is milk. These are 25 tea. And by doing so, he can avoid the contamination, because the special design for the flask is this angle of the neck. There's an angle for this opening, and what it does is that all the bacteria, any particle that is dropping down from the top will not enter in here. That's why he designed an angle like this. And the medium you should be kept underneath the neck. If you have medium that is run over the neck, it's very easy to get contamination because it's too close to the opening. That's why Dr. Carroll, he designed this flask to avoid the contamination in the cell culture. Okay, so this is the first cell line. So the first culturing is the chicken embryo. That is an organ culture. The first cell line is from human cervical cancer. By this Dr. Gay, he got part of the tumor tissues from this lady, Dr. Lex. That's why you got HeLa cells. Yeah. He, HeLa, is coming from her name. So she's the person. Did she agree to give us the cell to do the experiment? I'm not too sure about that. But she didn't know that her cell is like very popular these days. So Dr. Gay kind of isolate these tumor cells from cervical carcinoma, and then to do the culturing in a chemical defined medium. Now we call it DMEM, the Eagle medium. And he also he did this experiment in a sterilization condition in order to reduce the contamination. So we will talk about what is sterilization. In 1950, these all happened in 1950. The first insect cell were also appeared, but I forgot which doctor or which scientist isolate this insect cell. It's called the Grace cell. I forgot who did that. So that was happening in 1950s. In 1970, a lot of progressions are made, but these were all about procedure or the protocol to refine the procedure, to refine the protocol. For example, they use cell immobilization and develop suspension culture, or you can use a serum-free medium to culture the cell. And also develop the plastic ware. These are coming from the 1970s. Before 1970, all the cell culture are cultured in the glassware.
Wow.
Because we can do autoclave, high temperature, high pressure, under high pressure. using the plasticware, you have to use different way to do the sterilization. Now you can't put it into the autoclave because it needs to melt. Yeah. The disadvantage using glassware is washing, cleaning. Every time you use detergent, and if you have the residuals particle from the detergent, it will affect the cell morphology because it's going to dissolve your cell membrane. The cell will die. That's why the scientists decided to use the plasticware for the culturing. All you have to do after culturing, you don't have to wash it again, just throw away. Yeah, that's plastic. Okay. So that's happening 1970. Let's talk about the different culturing. Organ culture are take part of the organ. It doesn't have to be the whole thing, part of it. And then you can just grow in the medium. So what is in the organ? You have a lot of different cells. So it's not a single cell type. Okay? It's not a single cell type. The second characteristic is the organ needs a lot of different physiological support. Without the support, the growth of the organ is very, very slow. Okay? The most popular organ culturing is the umbilical culturing. Yeah, from the baby and from the pregnant woman. And umbilical culturing, all you have to do is cut the umbilical and put it into a medium. The cell in the umbilical, stem cell, endothelial cell, blah blah, will migrate out from the umbilical. And you can have, you will see, yeah, but the cell die in about a month. Everything die in about a month. Okay. Cell, is that a cell culturing? The second one is cell culturing. Where is, okay, I forgot to put the tissue culturing. Let me see. Yeah, I forgot the tissue culturing. For some reason, I delete that top one. The second one is the tissue culturing. Tissue culturing is coming from one specific part of the organ. For example, if I want part of umbilical, all I need is the outer layer of the umbilical. And you can do the tissue culturing. If I wanted to culture my skin, I can just put the top layer or the dermis layer and epithelial layer of the skin and do the culturing. And that's tissue culturing. For cell culture, you can only have one single cell type. For example, I can only have epithelial cell or the fibroblasts underneath my skin to do the culturing. That's one single type. Okay? For cell culturing, there are 3 types of cell culturing. Another 3 types. One is primary culture. Second one is cell strand. Third one is cell line. Do you know what's the differences among these 3? You probably know about what is primary culture, right?
Yeah.
Freshly taken from your body and then cultured. That's primary culture. And what's the cell strand and what is the cell line?
Maybe I forgot.
So cell strand and cell line are very similar. Yeah, oh, okay. So this is the primary culture taken from a blood vessel. So you can see all these, the round shape. These are monocytes. In the blood circulation, we call it monocyte. As the monocyte migrate out the blood vessel into the tissue, it becomes macrophage. For example, after stimulation by inflammation or by bacterial infection. So this is the primary culture taken from the blood vessel. Primary culture are derived directly from tissue of a living organism. A cell strain, this is a cell strain. A cell strain are derived from the primary culture. So for example, I have that blood vessel, and if I want just the monocyte, I can take the monocyte from the primary culture, right? That monocyte will become a cell strain. So this is the monocyte. This is TIG3. It's a fibroblast from the embryonic lung. Fibroblast from the organ lung. And what happens for this cell strain is one single cell type, but you can only pass for probably about 40 passages. Then this cell will die, will get old and die, and apoptosis, and you cannot do the experiment anymore. So what does that mean by 50 division or 50 generation? So if the cell double every 24 hours or every 48 hours, for a 50 division, I mean, you can only work on this cell for 100 days. What happened to your thesis? You'll never get graduate, right? You don't get to work on the same cell line for a long period of time. So what you want is something that could work for at least 1 year or 2 years in order to do a full research. So for the cell strain, the disadvantage for the cell strain is that you cannot culture for more than 50 division. That is about 100 days.
Can you do a subculture? So like to keep the
subculture, for every subculture is one division.
Oh.
Okay? What happens is if you have 100 cells in here, after 24 hours or 48 hours, you will double. So everything is all squeezed in this small tiny passage.
Yeah.
And keep that in mind. In vitro cell culture, all the cells are monolayer.
Yeah.
If you started to build up the monolayer culture, the cell underneath, at the very bottom of the dish would die due to the hypoxia.
Yeah.
And also, it's not anchorage independent. Therefore, those cells on the top will die too. So you have to do the subculturing, meaning split this dish from 1 to 2. So you have 100 cells in here. After 48 hours, you have 200 cells. So you will take everything from here and then spin down, concentrate, and divide into 2, and put one part in here, one part into another plate. That's called subculturing. For every subculturing is one division. Okay? For every subculturing is one division. And cell lines are single cell culture that undergo, it must have genetic changes, allow them to divide indefinitely. So they are immortalized. That means they live forever. Most of the time, the easiest way is to introduce some genetic lesion to disturb their cell cycle. So there will be no break for the cell cycle. The cell cycle keep going on and going on and going on. And the most common or most popular manipulator or the factor that will introduce into the cell strain is P53. P53 is a tumor suppressor gene. So what we do is we infect a virus large T antigen. It will bind to the P53. So P53 won't set the brake on the cell cycle. Therefore, your cell line will grow forever. That's what we do most of the time. And most of the cell line are derived from the cancer cell, because most of these cancer cells has the abnormality in the cell cycle, and we don't have to do anything. They just, they have indefinitely cell cycle going on already. So that's the difference between primary cell, cell strength, and cell line. Okay, let's go on the second part of the culturing. So this is a cell culture room when I was in the hospital, and I took some picture. This is the hood. This is a culturing hood. For a cell culturing room, you should have UV light in the room. You have UV light in the hood to kill all the possible bacteria, right? But you have to be very careful with this UV lighting. You can only turn on 30 minutes before you're doing any experiment, and you have to turn it off. What happens if you keep the UV light overnight? It's going to shine directly on all the surface, right? Everything gets sterilized, right? But it causes the metal or the wood become very fragile, and for a period of time, what happens is you touch the surface of a table, it kind of crumbling out. Yeah. Wow. So don't turn on the UV light too long. It's damaging a lot of stuff, especially the plasticware, because we put a lot of equipment inside the culture room. So if you turn on the UV light every day, overnight, I guarantee in 1 month, you will see all these paints are falling off from the incubator. You have
like this one?
Paint.
Oh, yeah.
Because it's kind of plastic. In the culture room, you usually have a hood and an incubator, and also the cylinder for the gas, CO2. And we usually have refrigerator, but I keep my refrigerator outside the culture room because I don't like the refrigerator. It has a lot of undefined space or area underneath the equipment, and that could have cockroach, insect, blah 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 hiding there, and I don't like that. A hood will be easier to clean than the refrigerator. And also you should have a liquid nitrogen tank for long-term storing. In our lab, we have a liquid nitrogen tank, but I don't use them anymore because students always, not usually, always forget to refill the liquid nitrogen. What happens if you don't refill the liquid nitrogen? It evaporates really fast. Every time you open the tank, the liquid nitrogen evaporates. And if you don't refill it, on the top of the cell will have temperature increasing faster than the bottom of the cell, and those are not stored properly at all. So nowadays we put our frozen cell line in -80 refrigerator. It's more secure than the liquid nitrogen, but it's not like the temperature in the freezer is -80. The temperature in liquid nitrogen is -140. That means if I can keep my cell properly in the liquid nitrogen, this cell can live longer than those cells I put into the minus refrigerator. Okay? So every 2 years at least, I have to renew all my cell line. Otherwise, they will die. That's the trade-off I have to take by using freezer. Okay. And we also need a microscope to observe our cell. There's one more thing I forgot to put in there. A centrifuge. We need a centrifuge to spin down the cell. But you have to keep in mind, don't put the centrifuge next to your microscope, because when you are doing the spinning, the surface of your bench is a little bit shaky. If you are looking at the microscope, you can't focus at all. So keep that in mind. Don't put the centrifuge and microscope on the same bench. You have to put next to it.
Yeah.
Okay? Don't put it on the same bench. So basic sterilization technique. Before you start to do the experiment, you have to wear your lab jacket, put your glove on, and spray the ethanol. 70 or 75% ethanol will be enough. Do not use 100% ethanol, because there is no water in the alcohol.
Yeah.
Without water, things that sitting on the surface will not dissolve out. So you need at least 70 to 75% of ethanol. You can also use 50% of isopropanol. It's equivalent. Okay. And so remember, you have to spray the alcohol on both of your hands, including interdigital spaces. That's very important. If your lab jacket, the sleeve is too long, please just roll it up, because that's the most dirty parts of you when you are doing your cell culturing, and that's most of contamination coming from
the sleeves.
From your lab jacket. And do not put any fresh plants or flower in the lab you are doing the culturing. All the fungus spore will attach to your lab jacket without your notice. It's very easy to get contamination. Okay, so you won't see those stuff in my lab. This is one of my students. She's working in the Zhangji, Zhanghua Christian Hospital. Yeah, she's still working in there. She works in the cancer center. She's doing, I don't know what she's doing. So the first thing to do the culturing is to set up everything in the hood. Remember, there is a shell or a glass shell. Sometimes it's a plastic shell that is separate the person and whatever inside the hood. Okay, so before you start to do the experiment, you have to lift it up and make a space for yourself. Do not open the shell all the way up, because you are giving the hood the risk having all the particles from outside and fly inside. So all we do is you can open about 15 centimeters to 20 centimeters. Okay, enough for your hands, both hands, to work in the hood. About this.
About 15.
Yeah.
You can imagine.
But don't get too concerned about, if I open it to like 50 centimeters, would I get contamination? No, because the hood has airflow in here. So you have air blow from behind of the hood, and then the air sucking in from here, or vice versa. So there is an airflow inside the hood that becoming an air shell, so preventing the particle flying in. Okay? So these are the tissue culture plasticware. We have the flask, we have the dish, culturing dish. And then, what do you call it?
So you can pipette.
These are pipette. This is the pipette egg. Pipette
Gun.
8. A-I-D.
Oh, I think, yeah, we call it this one.
Pipette gun?
Yeah.
Okay. And then we have at least 2 different types of pipettes. It's 10 mil or 5 mil. Internationally, 10 mil is all in red or orange, and the 5 mil is all in blue. And sometimes we'll have 1 mil pipette. It's in yellow. Sometimes it's in yellow. Oh. Once in a while, you will see a 25 mil pipette, and it's in purple.
Yeah.
Okay. And also you need, these are 6 wheels fast wheel. And these are 96 wheel for doing toxicity experiment. And also the same apparatus or the same plasticware for ELISA. You can code protein mill. Everything in here should be sterilized. And you can also have a smaller dish like this, 15 mils conical tube, 15 mils conical tube. So in order for you to get comfortable working on cell culture, you need to know how to transfer from the tube to the dish, right?
Yeah.
Without contamination. So can you put this down like this? No. Can you put this like this? Everything will fly into your tube. So you need to practice how to open the conical tube with one hand. Take your pipette, add in your pipette from here, and then you have to screw it in and put it in the rack, and then you add stuff in here, right? That's what you have to practice each day. So you can, play with this, and then I'll continue my stuff. So these are pipette, and here is the 25 mils pipette, and the pipette head. So the pipette head has at least one is up and down. It's very easy, and you can adjust the speed. Sometimes if you set the speed too fast, all the liquid will splash up or splash out.
And it will be causing contamination.
Exactly. We don't want contamination. The culture medium, we usually use DMEM. You can buy in a liquid form, and you can buy as a powder form. Okay, liquid form is easier, but it's more expensive. For a 500 mils, for a liter's DMEM, it costs about $8,000. NT dollar. So
1 liter.
Yeah.
8,000.
It's very expensive.
So expensive.
And the serum is also expensive too. I know serum, 500 ml serum costs about 10,000. And the antibiotic is the least expensive one. I forgot how much it costs.
Yeah.
So usually you will put like 10% of the serum in the medium and put minimum amount of the antibiotic. But too much of the antibiotic will cause restriction in cell growth. So just put appropriate concentration of the antibiotic in here. Usually when you bought this bottle, it's like 100 100-fold concentrated liquid, and just dilute 100-fold to your working solution. Okay, and then the culture media, if you buy the culture media in terms of a powder form, so you have to make into a medium, liquid medium, right?
Yeah.
After you prepare your medium, you need to sterilize your culture medium. Do not autoclave it. That means you are cooking the milk.
Yeah.
Think about cooking the milk. Everything aggregate, crumble.
Yeah.
Right?
Because there are a lot of proteins.
So for the cell culture, all the liquid medium sterilization is by passing through membrane, the 22 microns membrane. Okay. So there are a lot of different type of membrane. You can buy a huge flask membrane and then passing through from the top to the bottom. Or when I was undergrad student, I prepare at least 40 liters medium from powder, and my filter is about this big. It's a huge filter unit. Yeah. Okay. So you can start to do your try to practice when she takes you to our culture room. You can start to practice. First of all, you have to mark your own flask or your own plasticware. So you have to mark what is this cell line, the name of the cell line. Then is the day you seek the cell. For example, today. Then you have to put your name's initial. Very important is what passage is this cell line. Could be the 19th passage or it could be the 80th passage. The number of passage is you have to keep recording the number of the passage, because we don't want to do experiment by comparing a passage from 20 passage with the 80 passage. Okay. Metabolically, genetically, they could be different. Okay. So she's trying to take media into the flask. So she's trying to do this. So tight. Try to open the cap without touching the neck. Or you can practice with this conical tool. Try to open the large enough for you to put stuff in there, right? Try it. Like this. I have small tiny hand. You can do it. Here, real. Yes. It's okay. It's reuse one. And yeah, you have to go up a little bit.
Yeah.
Right. And no. Try to use one hand to close it. That's something for you to practice.
We use this one here in the lab though.
You don't do that in regular molecular biology experiment, but we do that a lot in cell culture. Okay, so she's taking, if you have, this is the tube, right? And this is your flask, and she's trying to open the bottle that containing the medium. What happened is her hand is over the opening of this flask. Yeah. And so that will cause contamination. That's sometimes just forgetting. Okay. So sometimes you can use this is a Bunsen burner, but we use what is that called?
Which one? Yeah.
What do you call this?
Oh. It's like we just call it Bunsen burner.
No. The Bunsen burner is this one. This is a Bunsen burner. That means it's coming from the gas. Okay, and this one is coming, just alcohol, burning the alcohol. What do you call those?
Spiritus. Spiritus.
I have no idea what to use. We use Bunsen burner in the United States, but we don't use those in the United States because it's kind of dangerous too. What happened is when the fire keep going on, the temperature increase. Everything inside this container will expand out, and you will have alcohol kind of spill outside this container. And what happened, without your notice, you're gonna get fire. Boom.
Alcoholic burn.
Yeah.
Yeah.
Alcohol burner?
Lamp.
Lamp.
Lamp burn.
Lamp burner. Oh. Okay. So when you work with this lamp burner, be careful. Don't let it sit there with the fire for more than 30 minutes. Otherwise, you're gonna see some alcohol spilling out outside of this lamp. But in my lab, we use pink alcohol. So you're gonna see if it spilled out. You're gonna see the pinkish stuff.
Yeah.
And then you have to observe the cell morphology under the microscope. So you have to observe how does it look like and what is the density of the culturing. So this is called 100% confluence. Everything is covered, okay? So what do you call about this one? If this is 100%, what about this one here?
50% confluence.
I would say this one is 50, so it's probably 40.
40.
And that's probably 20.
I see.
Okay? And the reason to know exactly the percentage of the cell density is that you have to know whether you need to do the subculturing next day or not. Okay? And most of the fibroblast or adherence cell has this spindle-like shape. If the cell density is very little, you will see the spindle shape of the cell because the cell try to get connection with its neighbor. That's their hand. Okay, so these are a couple commonly seen cell line. These are mouse 3T3. This is a fibroblast. Okay, this is confluence, and this is in very low density. This is a human intestinal cell line. This is vascular smooth muscle. This one is smooth muscle. This is the monocyte. That EC cell. I forgot what is that. Is that endothelial? Endothelial cell? I forgot is that endothelial cell. I forgot what cell type is that. Okay, so you have, after you observe the cell morphology, you have to count the number of the cell you have. There are a lot of. Oh no! What happened? Copy and paste. Fail. So we need to know exactly what is the density or how many cells do you have in this culture. To count exactly the cell number will allow you to calculate what is the doubling time for this particular cell line. That means how much time does this cell line take to get from one cell to two cells. Okay, that's doubling time. So in most of the lab we use the hemocytometer. This is a hemocytometer to count the number of the cells. In a lot of clinical lab, they don't have time to count it. They have to use the culture counter. I mean, you put it into a 15 mils conical tube and put it, there will be an electrode, and then you can measure it. What happened is cells in here are contaminated. You cannot put it back to the culture anymore. Okay? So that's the disadvantage of using culture counter. So let's look at this hematocytometer. It's a, I forgot to bring it. It looks like a slide, a regular, the size of a slide, and it has. Let's see. Do I have? This is like size of a slide, and it has grid on the top of the slide. And what it does is it looks like there are a lot of line in here, making it 9 different sections underneath the microscope. In the higher power, under the higher power microscope, you can count the cell on each section. For every 1, 2, 3, 4, 5, these bigger section, you have 5 and 5 cells in a row, so you total have 25 cells in one section. You have to count number of cells on the top of the grid, and keep that in mind. Each section should not have cell more than 100. If you have this too many cells that cluster in here, sometimes your counting of the cell number will not be accurate.
Yeah.
So if you count it per square, is like 200. Try to dilute it 3 times, okay, into 1/3. And from here, you can count. You can, if you know your dilution factor, you can calculate what is the total number in that in your concentrated cell. Okay. So when you're doing the whole practicing, subculturing, she will tell you how to do this. After you count the cell, at the same time when you're counting the cell, you wanted to know how many cells in this culture are still alive, how many cells in this cultures are dead. So how can you tell the differences between a cell is alive or dead?
We're using the assay, MTT assay.
Yeah, so you know the MTT assay.
I know.
You know, do you know? MTT assay is testing the enzyme inside the mitochondria, whether it is active or not, by sending it a compound. I forgot what's the name of the compound. MTT
compound.
And if the enzyme inside the mitochondria is active, that means the cell is alive. This compound MTT, a purple, is that purple? No, it's a yellow. It's a yellow solution. Will be converted into a purple crystal. Okay, so MTT is a assay like testing whether the cell, the testing the enzyme inside the mitochondria in order to know whether the cell is alive or dead. It's expensive, take a lot of time. It's not convenient. So normally we use the trypan blue stain. Trypan blue is a small compound, a blue dye. What it does is that it will be, when you mix the dye, the solution with the trypan blue dye, if the cell is alive, you will uptake the dye by pinocytosis. I mean, your membrane integrity should be okay, and then you will allow the dye going through the cell and let it go. So no blue particle will stay in the cell because it's actively taking up and let it go. if the cell is dead, the membrane, the integrity of the membrane is not normal. I mean, you probably have hole in there, so all the dye will go in there, and all those blue particles will stay inside the cell because no vesicle, no mechanism will try to push it up. So you will see a blue color in your culture. So you can count the cell and see whether they are alive or dead on the hemato cytometer. So if the cell is alive, you see this white spot. And this is a dead cell, and this one is a dead cell too.
Yeah.
So you have to count it. By counting the number of the live cell and dead cell, you will know your total cell number and the percentage of the survival. Okay?
So in the Trypan stain, basically the dead cells will obtain the blue stain,
yes,
but the
yes,
alive cells will push it
clear,
out.
yeah,
Okay.
be a clear one. That's
Can
a
I
viable sign.
why the fiber cell will push the Trypan blue out of the cells?
Think about fluid goes in and goes out. Your a normal cell, you will pick up the fluid and it will secrete it out too, just like a vesicle transport is normal. That cell do not have the system going on. So, and also on the top of that is the membrane integrity is bad. So every blue particle will go inside the membrane and get really, really blue. It should not have pinocytosis or other transport system to uptake the medium inside the cell, but the membrane has a hole in it. Therefore, the blue particle goes in there. The different blue particle, if you try to run a SDS page, you will be at the bottom of your gel. That means the compound is really small, very, very small. Okay. Okay, this is the growth curve of yeast and the cell line. So you all know the growth curve of the yeast, right? The lag phase, exponential, and the stationary. The cell line growth curve is a little bit different. The lag phase is not so obvious. Okay? So you will see something like a small hump, and then it will start to decline. The phase for the declining is more is sharper than the lag phase for the cell culture. That's the difference between bacteria or yeast culture and regular cell culture. And that's a different cell line will have different doubling time. Okay, just keep that in mind. At the end, after you do packaging and maintaining a lot of the cell, you wanted to, or after you do your experiment, you want to freeze your cell. And freezing cell, first step of freezing cell is to remove your medium. And most of the cells are here at the bottom of your plasticware, right? So what makes the cell get attached on here? What? Now the medium, I remove the medium. I can wash it with PBS. The cell is still here on the top of your plasticware. So what makes this cell adhere to the plasticware?
Adherence protein.
Protein, adherence protein,
Yeah.
correct. There are a lot of different types of protein that secreted by different type of the cell you are culturing, and you will adhere on the plasticware. So all you have to do is buy enzyme, trypsin, because it's really cheap. Okay? And it's not specific cleaving anything, not including your membrane. So you wanted to culture your cell with trypsin medium in very short time, less than 10 minutes. Otherwise, it's going to damage your membrane. All you want is to and then you concentrate your cell, store the cell in 5 to 10% of the DMSO. Why? You can store it into glycerol too.
Oh.
So why you wanted to store your cell in DMSO and put it into liquid nitrogen?
Because DMSO has the ability to prevent as a bioprotectant to prevent the
Ice
the
formation.
freezing. Yeah, the ice formation.
Ice crystal formation. Okay, and by using DMSO, you prevent the crystal formation. Also, we don't want it to freeze the cell right away. If you freeze the cell too fast, from room temperature to minus 140, the cell did not get the time to exclude out the water, and so you'll die right away. We wanted to do it gradually by, dropping the temperature 1 degree per minute. It's very hard to do that. So what we do is we put the concentrated cell in DMSO, remove it from room temperature and put it into 12 degrees refrigerator for 4 hours. Then we put it into the minus 20s freezer for overnight. Then the second day we put it from minus 20s freezer back to the 80s degrees freezer.
Oh.
And then you can store it for at least 3 or 4 years. And after minus 80, you can put it into liquid nitrogen, and will stay there for at least 10 years. I'm not trying to stay here for another 10 years. So I use minus 80's freezer. Okay. So after you do all your experiment, remember to clean it up. Keep your workstation, your surface clean is very important. So normally we clean up the hood at least once a month. You apply the detergent water first. Wipe it up with water and wipe it up with bleach. Water first, then bleach to kill all the bacteria or fungus, especially the fungus. Then the bleach is very hard to clean it up. So you need to apply at least one detergent. For example, SDS or 7X detergent is the best. And what it does, it will lift up the bleach crystal that sit on the top of the surface. Then you have to apply 3 times water. And then finally spread, your ethanol. That's how we do a thorough cleaning for the hood. But normally, every time you finish your experiment, you just wipe it up and spray with ethanol. That's it. Okay? Just keep it clean. A lot of laboratories, they will leave equipment that needed for doing the cell culture. For example, your pipette and your tip, blah blah blah. You see them inside the hood. I don't like it because it's the same thing that usually we have to turn on the UV light, right? What happens is it shines directly on all these plasticware. And if you keep this plasticware inside the hood too long, it starts to have little crack on the top. And you will not know that. You won't know that at all. So it will get contaminated really easily. So we don't do that anymore. We only take necessary stuff inside the hood every time we do the experiment. When we finish the experiment, we take them out again. That way you don't even have to turn on the UV before you do the experiment. Just keep that space clean. Okay? And that's it. So next you guys need to discuss when you're gonna start your experiment, okay? So I put, for your first experiment, you have to throw in the frozen cell. You have to start your own culturing. And then you have to do the subculturing. That's all you have to do. But when you're doing the subculturing, you need to count how many cells you have before you subculture.
Yeah.
And then how many cells you have after you subculture and after a couple of days after, per generation. So what is the doubling time? That's why I want to ask you. I put the protocol in the cloud already, but these protocols are it's the protocol that when you first do the experiment, you have to follow, the flow. But
Yeah.
in our lab, sometimes we skip certain steps in the protocol because it's not necessary.
Yeah.
But I wanted to show you what is the proper way to do the experiment first. And in our lab, sometimes we don't do that at all, okay? But proper way is right here.
This also happens in our lab.
It happens in every lab.
Yeah, we skip
Yeah.
most
You
of
skip you skip certain step of the protocol.
Yeah.
For example, the protocol asks for washing the palette for 3 times. You're probably gonna wash 1 time.
Yeah,
관찰했으면
I understand.
Yeah. Especially in the cell culturing, we don't want it to wash too many times because it damages the cell. Every time you wash it, you lost a lot of cell again. It's not a very good protocol at all. But that's the proper way to do the experiment. Okay. Okay. That's it for today. See?